Hello, this is Dr. Lucy Abkarian, course Bio 225, Genetics, lecture number 4, chapter 3, part 2. Here we show the classical independent assortment of gene, which means that prevalence of one gene does not alter the presence or expression of another gene. Here we show two genes, big A and big B, each with two alleles. We can have 4. The segregation of these two alleles can yield 4 different outcomes with total of square of 4 or 16 different genotypes that result in 4 different phenotypes in F1 progeny. Of course, it goes one step deeper when we begin talking about F2 progeny, in which genotype is segregated 1, 1, 2, 2, 1, 1, 2, 2, 4, as shown in column 2. Here is an assignment question. Please write out the genotype of each individual in the F2 population. We may observe a seemingly ungiomatous phenotype in the F1 and F2 progenies when there is a gene interaction, but there is nothing mysterious here. When we have parental lines such as big A, little b, and little a, big B, our F1 will always have big A, big B, little b genotype and the associated phenotype. From this point on, the F2 will have the characteristic 9, 3, 3, 1 phenotype that we see in independently segregated genes. Homework research assignment. In contrast to independent segregation, what is a dependent assortment and what might be the mechanism? Such distribution will be observed only when the dominance of alleles at both genes is complete. Please remember that the 9:3:3:1 ratio is a statistical number. The actual number will come close, but most likely will not be in exact proportions. Complementary gene action in sweet peas. Let's now examine how purple F1 progeny are produced by crosses of two pure breeding white lines. It may sound strange, but it's quite straightforward. Pure breed parental genotypes big A, little b crossed with little a, big B will yield all identical F1 progeny of big A, big B, little b. In this case, the genes are complementary, and when they come together, they will express a trait that is not present in pure-bred parents. In this case, purple flowers. The F2 progeny will produce whether white or purple progeny. All F2 individuals that carry either AA or little aa or big BB or little bb will produce wild flowers, while every other genotype will produce purple flowers. In this case, the phenotype ratio of dihybrid F2 protein will be 9:7, in which 9/16 will be purple, and 7/16 white. Now, let's consider the possible biochemical basis of the complementary gene action. It is possible that genes big A, or big A, little a, where A being dominant over little a, codes for a functional enzyme big A that catalyzes first stem in synthesis, but this process will not be completed without the presence of enzyme big B, which is coded by big B or big B little b gene. If we have little a, then we lose the function of enzyme big A. If we have little b, then we lose the function of enzyme big B, and no purple pigment is produced. The purple is produced only in the case of big A blank, big B blank, which explains the phenotype observed. Epistasis is the phenomenon in which an allele at one gene masking the effect of another gene. The gene that does the masking is said to be epistatic to the other gene. Conversely, the gene that is being masked is said to be hypostatic to the other gene. Just like in other cases, epistasis can be recessive or dominant, and just like in classical cases, recessive epistatic gene must be homozygous recessive. For example, e, and the dominant epistatic gene must have at least one dominant allele present. For example, big E. Let's examine the recessive epistasis in golden Labrador dogs. We observe 9:3:4 ratio in F2 progeny of dihybrid crosses, which indicates recessive epistasis. 9/16 black, big B, big E. 3/16 brown, little b, big E. 4/16 yellow, big B, little e, little b, little e. As you can see, in 25% of the F2 progeny genotype, little e masks the effect of all big B genotypes. Here are some examples of epistasis. Recessive epistasis in humans with a rare blood type, Bombay phenotype, about which we have talked previously. Here is an example of dominant epistasis 1 in summer squash, where the dominant allele of one gene masks both alleles of another gene. Here is an example of dominant epistasis 2 in chickens. In this case, the dominant allele of one gene masks the dominant allele of another gene. Now let's summarize what we have discussed in this chapter. Observing the F2 ratios below is diagnostic of the type of the gene interaction. These F2 ratios occur only in dihybrid crosses where there is complete dominance. So, gene interaction type, none. 4 distinct F2 phenotypes. Example would be lentil, seed coat, color. F2 genotypic ratios from F1 dihybrid crosses will be 9, F331. Gene interaction complementary type, where one dominant allele of each of two genes is necessary to produce phenotype. The example case would be sweet pea, flower color. Ratio would be 9:3:1, F2 phenotypic ratio 9:7. Gene interaction type recessive epistasis, where homozygous recessive of one gene masks both alleles of another gene. Example was retriever's coat color with F2 genotypic ratios from F1 dihybrid cross 9:3:1 and F2 phenotypic ratio 9:3:4. Gene interactions type dominant epistasis. 1. Dominant allele of one gene hides effects of both alleles of another gene. An example was the summer squash with 9:3:1 ratio, F2 phenotypic ratio 12:1, and dominant epistasis 2, where dominant allele of one gene hides effects of dominant allele of another gene. That would be the case of chicken feather color with 9:3:1 ratio from F1 dihybrid cross and the F2 phenotypic ratio 13:3.